Hi, this is Dr. Ben Morrill. Welcome to episode 49 of Reptile Genetics Weekly. Glad to have you here. And as always, we'll start with updates on tests and results going out to everyone. So now we have 18 tests that we can do as fast tests, and we'll list those out a little later in the episode. So those fast tests are going out multiple times a week still. You get 1 to 2 day turnaround once we get the sheds to us. So yeah, those are going out, I think, probably again Tuesday night and at least 1 or 2 other times this week. So and then for the next large run where we're doing full panels and the few tests we don't have as fast tests yet, that will be the first week in April. We'll run that, and then results probably coming out sometime in the second week of April. And as we get closer, we'll give you a more specific time. But that's it for sample updates. Kayla, how are you doing?
Hey Ben, I'm doing great. I am I'm doing well. Better than last week. Still looking like my hog nose is gonna have to have surgery, but she did end up passing one more infertile egg and eating another meal for me. So
there's 6 left.
6 left. Yes, she has 6 more eggs left in that oviduct. So your pushy thoughts are working. Just keep sending them to ask her. If you want,
we ask.
Yeah. If you want to hear the full story, go to last week's episode, episode 48. And we have a whole. You'll get the full saga, because it was a lot. But it's okay. She's doing great, and I mean, girl hasn't lost any sass at all. Like, I offered her mouse, and she went, and then just like slammed it into her bedding and like rolled it in there. I was trying to like get the bedding off of it before she started swallowing it, but she was like, "No, I want it breaded." So
shake and bake.
Exactly. So she is doing fine, I guess.
Wow.
So anyway, genetics wise, you've been working on some more tests. Well, you're Ben. You're always working on more tests. So what do we got?
Yeah, so we put this up a couple weeks ago. This is what we have coming. These tests we have listed here are ones that I don't know for sure what we'll get. As those of you that have been following for a while, you know that sometimes we get them on the first try. Sometimes it takes 3 or 4 tries, and several of them in this list, it's at least our second try trying to get them. But yeah, over the next 2 to 4 weeks, I should be able to start getting more and more information about all these in this list. And there's actually several more that we're shooting for as well. But yeah, every mutation is its own journey, and these are the ones we're hoping we'll be able to wrap up a good number of these.
Yeah, I know you've been working hard on TSK and GCR for oh gosh, it's been like 3 or 4 months now, right?
Yeah, well, like Monsoon Monarch, all 4 of those first 4 put a lot of work into those. But
we'll get there. All right, and let's see. So as far as non-ball python genetics, where are we at there?
Yeah, so we're still working on genomic registries, even for ball pythons. That's something that we're trying to get. That would be where you all would be able to send a shed in for your ball python, and we would do a whole genome sequencing and save that data. And then any new tests we get in the future, you can just let us know you're interested in the results for that. And we won't need another shed because we'll have all of your genomic information. So that'll be a really cool option once we get that. But yeah, besides that, it's also going to be helpful in Madagascar boas, the doom rolls, and then the ground boas, be able to have a purity test there. And for panther chameleons, we'll have locality tests. For Drymarchon, we're going to have essentially like a relatedness test, so you can make sure you're pairing animals that will have the best chance of giving you the most healthy babies. And obviously these same things will be applicable to other species where you have similar questions. So this is just the beginning of that aspect, moving away from tests that just look for a single mutation or something like that to where we look at everything and record everything and have that in a database that we can go to later. Whenever we have a new thing come up, then we can look at your information if it's something you're interested in and be able to give you that answer without having to extract DNA again. And so we're pretty excited for that. And that's looking like over the next 2 to 4 months, we'll have some significant progress there. Every time we we get some good progress, then we find out something else we've got to do, and then we make some progress and we find something else. And so it's a iterative process, but it's looking good.
We got this. Okay. So for fast testing, it is a much longer list than it was last week. So what do we have going on there?
We're at about 2/3 of the way, so you'll be able to see the list there, pause and look at it if you want. But starting Thursday this week, we'll have everything on this list where we will run it as a fast test. The only one, albino, we have down in the bottom in black. We actually found out that we had an issue with the albino fast test. So the customer or two that were affected by that, we've already contacted. But yeah, we're going to have to rework the albino one. And so it'll be another probably couple of weeks until albino will be available as a fast test again. But we still, as we have in the next slide, it's still looking like by sometime in April. Oh,
there we go.
Yeah, sometime in April, we'll have all of them. And then, I forgot we have this slide where we show the next few that will likely be ready. Probably in the next couple of weeks, we'll get red stripe, black pastel, spark spectre, orange dream leopard, and maybe some others if we get lucky and get them on the first try. But those are the next few we'll work on, and then after that, we'll get the last of the blue-eyed Lucies and the few other odds and ends. But yeah, it's going really well, and like I said, by sometime in April, we should have all of them.
That'll be great. So I know that we were talking earlier about new paper that is kind of tangentially related to the piebald gene, but it's not completely about that. So what's going on there?
Yeah, thankfully, like we say here, thanks to Dr. Heather Roffey, she made me aware of this paper. It's pretty cool. She was one of the researchers that did the piebald mutation, worked on that originally, part of that Canadian group we've talked about multiple times. But yeah, she let me know there was a pretty interesting paper that came out talking about the same mutation that causes piebald in ball pythons. So the researchers wanted to look and see as far as the chromatophores, so like we have listed here, those are cells or placids that contain pigment. So when we talk about axanthics and amelanistic or albino, erythristic with corn snakes. So those are all different types of chromatophores or cells in our animals that have colors. Well, what was really interesting is when you look at anoles, like we talked about in, I think, our second episode, where they put the same piebald mutation in anole lizards. And what they found was the normal anoles had iridophores, like the last one in that list there, that makes it more iridescent. So the normal anoles had iridophores, but the ones with the TFEC mutation didn't. And so that was a big difference, was that type of chromatophore is not present in the animals with the TFEC mutation. Well, the interesting thing is they also looked at adult ball pythons. Some were piebald and some were normals. And it wasn't like that with ball pythons. There were no iridophores in the normals, and there were no iridophores in the piebalds. And that was very surprising. They did not expect that at all. It seems to be in, pretty much any vertebrates. We're going from a zebrafish to an anole to a rat snake. They see this TFEC. When you knock it out, you lose the iridophores, specifically in the brown anole paper. But with a ball python, it doesn't matter whether it's the TFEC is there or if it's mutated, you don't have iridophores either way. So that's very surprising.
Yeah. And other than just like the rainbow color, it can also affect like the shininess, I believe, in like the sheen of the, I guess, the skin and scales. So that's interesting. So I'm not really sure as a layman, like what exactly, were they able to project, like, what about ball pythons might be causing this, or what the implications are of that? Or was it just a, boop, here's what we found?
Exactly. They're like, we don't know why, but there's no iridophores in these adults. Now, they did mention that not in the adult ball pythons. They did not look at any juveniles or any embryos. So it's possible that there are iridophores during some developmental stages, but by the time they're an adult, whether they're a normal or a pied, they don't have iridophores either way. So it was very surprising. So they talked about several different avenues to go down with CRISPR editing, with, you know, different things they can test and try to figure out, because that was definitely a surprise.
Yeah, because I mean, it's not like ball pythons aren't shiny or aren't iridescent in some light. So that's interesting to think about.
Yeah.
And so they, like, was it just the skin that they looked at? Like, did they take, because they took the animals and looked like in their skin to test for the presence of these different chromatophores. And I think the eyes as well, like they're, I think basically the things that we look for as breeders that produce color and your pretty phenotype that we tend to breed for. So.
Yep. Yeah. So, you know, I would imagine next they'll look at, you know, brand new babies and maybe even sacrifice some eggs and look at some developing embryos and see if at some point there are some iridophores, or maybe there's never iridophores. And then that was a really interesting thing. Just like we talk about with, you know, with the Barnhart black pastel and with yellow belly and Ultramel, where there's multiple mutations, even though they look the same. Whereas with, you know, gravel and asphalt, they do look a little different, and they're different mutations. Like there's stuff, you know, when you start digging around, that's an interesting thing about science. A lot of the time you go in with a specific question and you come out with like completely different questions, and sometimes more questions than you started with. This is certainly a case in this paper. They had one question that came out with something completely different that they didn't expect at all.
That's cool.
One quote, I think it was one of the professors I worked with, he said, really, all we do as scientists is prove what we used to think was wrong. And we just keep doing that over and over again.
Yeah, sounds right. I imagine the process is pretty humbling. But I mean, hey, it's fun. And I mean, especially when it has to do with reptiles. So.
Oh yeah.
That's cool.
Yeah, that's the exciting thing about this, is like one of the first sentences in this academic paper is it talked about reptiles, some of these pet reptiles becoming a model for looking at color and pattern, because we have so many different color mutations. It's really cool. Like 10 years ago, you didn't really see this at all. 5 years ago, you started to see it, and now we're you know, seeing it more often, which is really cool to see. Some of the things we're doing as captive breeders also being interesting to academic researchers. I love seeing that.
Yeah, I mean, they've, like, so many of these breeders, even just, you know, these old guys that, you know, as far as some of these, like some academics previously might have thought it was just some guy in a room with his snakes, which, yes, that is what we are. But you've been really, like, breeders have been doing some really incredible science with this, like, genetic selection that they've been doing for decades too. So it's cool, and you're right. I'm glad to see this this moving forward and being recognized in the academic world, because I mean, yeah, no, I'd love to see more of that being looked into. So cool.
Yes.
All right. So next, it is time for technically we did this earlier this month, or at least I think we did. But anyway, it's time for another monthly Morph Spotlight.
Yeah, we decided we've got so many morphs, and we've got more that we're developing tests for, that we probably have to do it more than once a month. Maybe every other week we'll do a morph update, and then the other week we'll do a why I do this, because we've got lots of morphs to talk about.
All right, sweet. So this time around, it's Ultramel. So go ahead and tell us about Ultramel.
So yeah, with Ultramel, like many of you that have been watching the videos, we've talked about this multiple times, but definitely lots of people, it's still a new concept. So with Ultramel, there's two different mutations, and we've kind of called them Ultramel A and Ultramel B. The first one is a simple base change that was in the original paper that Dr. Sadell and her students did the work for that. But when they did that publication, they knew there was another mutation that must be causing it, because sometimes you'd have a clearly Ultramel animal, so it's a visual Ultramel, we'd get the shed and do the test that she had developed, and it would come out as a het. And we know it's not a het because it's visually an Ultramel.
Yeah.
So what she was able to find out eventually is something I worked on too, but she was the one that eventually figured out what the second mutation is. And the second one, the Ultramel B, in the United States at least, is much less common. So the vast majority of them are Ultramel A. But Ultramel B ends up that it's not a simple, base change or, one or two base deletion or something like that. It's actually a bigger deletion, and there's some kind of swapping like inversions and things like that going on. So it's actually a fairly complex mutation. But as far as us as breeders, whether it's homozygous for Ultramel A or homozygous for Ultramel B, or het has one copy of each, they all look the same on the outside. So those changes, just like the yellow belly -1 and the yellow belly -17, both those yellow bellies look the same, and the ivories from them, as far as we know so far, also look the same. So in this case, Ultramel A, Ultramel B give you something that looks the same, but they are actually different DNA mutations.
Interesting. Okay, this one might be putting you on the spot slightly as far as, but you say a base change and an inversion. Like, is there a quick and dirty way you could explain, what that is?
Yeah. So like if you focus in on Ultramel A, so I don't remember the exact sequence off the top of my head, but say it's TTC. And right there where it's TTC, and that's what you see in a normal animal, you see two copies, both copies are TTC. In an Ultramel A homozygous animal, it would be TCC. So you have two different copies where one of the bases has changed from a T to a C. So it's homozygous for that C mutation. There's not a T on either side. Now, if you get one T, one copy says T, one copy says C, then that's a het for Ultramel A.
Makes sense. Okay, so it's just the T, T, and C are the kinds of proteins that make up part of that DNA chain, and when you have a different protein, it's
DNA sequence.
The sequence,
so, yeah. So that DNA sequence, a lot of the time, will make it so the resulting protein from all of the DNA sequence is different or completely knocked out. But right. It's specifically the DNA code itself just has one little change that actually makes a big difference.
Right. Okay. Cool. Thank you for explaining that.
And inversion is where you'll have like, it could be 20 or 30 base pairs, or it could be 500 or 5,000 base pairs. But where it's reading one way, there's a mistake that's made, and part of it gets cut out and flipped around, and then it goes back into the same spot. So now you're reading completely normal, and then you hit that inversion, and so you're actually reading like this, and then you're coming back out that way again. So it's just like this little piece got cut out, but then it went back in, and it's the wrong way.
Interesting. And so the fact that you have two different genes that make up the Ultramel mutation, even though you have one that's a base change and one that's an inversion, they can still be compatible and create the same phenotype or the same physical expression that we call Ultramel. That's really cool.
Yep.
So and this was another thing that Dr. Seidel did, right?
Yeah, so it was the same paper, the last Morph update we did, or Morph of the month we did with albino, and the same people that donated sheds, and we have that same list. So lots of people that we've talked about previously and now, and it's definitely cool. This was several years back. There's been people donating sheds to help out with this for many years now, and thank all those people once again for doing that.
So how did these tests shake out, since we have two different genes that we're testing for?
Yeah, so when you send an Ultramel test in, we actually do both tests. And so the Ultramel A1, since it was in that original publication from Dr. Seidel and her students, that one we've run thousands of times, and there's other people that have also run them many, many times. Dr. Seidel ran them many times. So that one's an A+. We feel very, very comfortable and confident with that one. Very high accuracy, over 99.9%, as long as there's not a human error with mixing up a shed. And then for Ultramel B, only by the fact that we haven't ran as many tests so far, it's been a great test. So it's a B right now. But by sometime this year, I expect to where we will have run over 1,000 known samples, and we'll be able to raise that up to be A plus, since it's part of. Actually, I guess technically there is no publication for Ultramel B yet. I forgot about that. So
Oh, okay. So it would technically just be an A.
It'd be an A, unless Dr. Siddell ends up publishing the specifics about that Ultramel B, that second mutation. So it'll either be an A or an A plus, depending on if Dr. Siddell publishes about it.
Mm-hmm. And that's kind of how we grade ourselves as far as like the accuracy of our tests, because we don't call it an A plus unless it has over 1,000. It's been tested over 1,000 times on 1,000 different individual samples and has a paper written about it. But if it has all those samples over 1,000 but no paper, then it's just an A. So kind of our tests are high achievers, ideally. So that's cool.
All right.
Now, Ultramel time over. Cool. Thank you, Ben. And now it's time for a shed donor shout out, which is a new thing that we've been doing. Like we'll throw out a thank you to people who donate a lot of sheds, especially when we introduce a new morph test. But we've decided that there are so many people that we may as well just start giving people a shout out on the show every week. So
yeah, I want to let Jessica and Tommy know. I say thanks. They're kind of the ones that brought this to my attention. There are so many people that have donated sheds, and we definitely can't do, like we've said many times on here, we can't do anything without sheds coming in, and we're very, very thankful. So don't be surprised if you see an email from Kayla sometime. If you're someone that's sending sheds, you might get an email from Kayla asking for some information, so you can potentially be in a future episode. But we definitely thank you very much. And Eric Chung was one of the very, very first people to start sending sheds to me very, very early on, and talked with him at Daytona. It's been probably 3 or 4 years ago before we really started doing anything other than colubrid sex determination. He's always been very supportive and made sure that he got sheds to us that can help out. And so, yeah, if you're interested in looking at some ball pythons, you can check out Eric Chung Reptiles. Thank you so much. He's been doing it for a while and has taken the time.
2017 says he's been on Morph Market since then, at least. He's probably breeding since before then.
Oh, yeah, he's definitely been doing it longer than that. And I know at least for a while, he may still be doing green tree pythons too.
Oh, cool.
But yeah, definitely thank you so much, Eric Chung. And like we said, we'll probably be doing this kind of like the morph update. We've got a lot of morphs to talk about, morph tests, and we've also got lots of shed donors. We want to make sure that we let people know who's taking the time to collect sheds and organize and send them in and give you a public thank you, because it means a lot to us.
Yeah. Also, it gets more eyeballs on your logo, which is a good thing for breeder businesses.
Yeah, sure.
So win-win for everybody. All right. And if you want to be a part of that, you can also, of course, when you send in sheds, but also send us an email, with your name, your logo, your Morph Market and website link, what you're excited about, pictures you want to share, and all that goes straight to me, raregeneticsocial@gmail.com. If you send it to me, it's less likely to get caught in the mix of like order questions and stuff. We do answer everything on there, but you know how hectic it can get. So, we'd love to have you be the next one in our future shed donor shout out. All right. So Ben, got anything else for this week?
That's it. Just thanks again. We're cranking away and excited for baby season. Have everything as a fast test, and
yes,
some more new ones coming, and to have those available as fast tests, either from the very beginning or very quickly thereafter, is going to be. We're very excited for how 2024 is looking.
Oh yeah, for sure. All right, guys. Well, thanks so much. Make sure you like, subscribe, comment below, hit the little bell so you get a notification when we post something. It works. It works like 80% of the time, every time, as far as I can tell. So yeah, thanks guys, and we'll see you next week. Outro in 3, 2, 1.